Good day once again. Welcome to today's class on genetics. It is true, it is basically for medical students, yes, but we will learn a lot from it as medical students and as non-medical students. If you are new in my channel, you're much welcome once again. Please feel free to click subscribe button, click like button, turn your notification button on. Feel free to do that and feel free to share it with your friends and classmates. Okay, let's kick off. Genetics is a popular word, so to speak. Let's see what it means. The word genetics was derived from Greek word gene, G-E-N. This is a Greek word, and this word means birth or origin. It means birth or origin. This genetics, which was derived from the Greek word gene, it was derived by a scientist known as William Bateson. William Bateson in 1905. So we've been able to know where the word genetics was derived. That is from the Greek word gene, and it means in English birth or origin. And we also said it was derived by a scientist called William Bateson in 1905. It's also important to know who is the father of genetics. Answer: Gregor Mendel. Gregor Mendel is the father of genetics. Gregor Mendel is the father of genetics. This man or this scientist we called Gregor Mendel was an Austrian monk. Austrian monk. Yes, he was an Austrian monk, and he used a garden pea, a species of garden pea called Pisum sativum. A species of garden pea called Pisum sativum to carry out his research on genetics so as to get all the findings, all the results we have today. Pisum sativum. As the class continue going on this genetics, we will see in the long run why this scientist decided to choose Pisum sativum to carry out his experiment. Okay? Now, since we have noted where the genetics was derived from, the scientist, the year, and the father of genetics, now let's see meaning of genetics in a broader way. Genetics can simply be defined as the study of how gene is transferred from parents to offerings. Offerings simply mean children. How gene is being transferred from parents to offspring. You notice that if parents are fair, you see the children being fair. Sometimes you see the children being dark. You see the children having pointed nose and all have you. It is gene that is responsible for that. So it studies how gene is being transferred from parents to offsprings. It's important to also know that there is another word we call genomics. Genomics. What does it mean? Genomics studies all persons' genes. All persons' genes. It studies the interactions of one gene to another, or interaction between one gene to another. You also study the mutations that could occur in a particular gene. So you now see that genomics study a broader thing. The interactions that occur in the gene with another gene, the mutations that occur in the gene with another gene. In case you don't know what I mean by gene mutations, it simply means the sudden changes that occur in gene. So we say that genomics studies it. So that means when you are comparing gene and genomics, you say both are derived from the Greek word gene because they are talking about gene, the study of gene. Yes. Now, the difference there is now that genomics will study a broader thing, a wider thing. The interactions that occur between one gene and another, the mutations and all have you, it will study it. Yes, why we say that a study interaction and mutation is that sometimes we do experience interaction of one gene and another, or between one gene and another. We do experience or we do see cases of mutations in the gene. Yes, take for instance, it's possible for a particular part of a body to start developing or start growing a tumor or have a growth of tumor in that part of the body. Take for instance, when someone's breast is developing tumor or develop tumor, that is mutation. Mutation has taken place. Yes, you know that we have 10,000 genes. A woman body has more than 10,000 genes. Each or any change, any mutation that will occur in that one gene can affect other gene. Yes, take for instance, if I have a or if someone have a mutation or outgrowth or tumor develop on the breast, it is gene mutation. There is already sudden change in that gene on the breast. This tumor that is developed is located on a portion of a body, on a particular organ or tissue, so to speak. It's located on a particular organ or tissue. So we say it is localized. Localized. That is, any tumor, any growth on a particular point of a body is said to be localized. That is benign tumor. Benign tumor. But if this outgrowth or if this tumor spreads round other part of the body, if it spread round other part of the body, it will no longer be localized. We will call it at that point malignant tumor. That means it developed or is developed to what? Cancer. It's not a cancerous cell. It has spread around the whole body. And in such case, to treat it is not an easy thing. It's not an easy thing. So when the person is rushed to hospital, we have rationale we carry or we follow to treat such person. We want to understand what we mean by malignant tumor or cancerous cells, malignant cancer. We want to understand it fully. Try to watch a video on Heller cell line. Just type Heller cell line online. This is one of the important cell line we have in the field of medical or medicine. Heller cell lines. This cell line was gotten from a woman known as Henrietta Lacks. Henrietta Lacks. Yes. So when you watch the video, please feel free to comment or chat with the comment box so that we'll interact more on it. Heller cell line. Yes, you now see how he developed malignant cancer or malignant tumor which spread around the whole body. And let me not gist you all about it, because if I gist you will not be willing to watch it. Watch it for yourself and see why it is a popular cell line noted in the field of medicine. Yes, that is it. I've been able to explain that if the tumor being developed on a part of a body is only on a particular part of a body, maybe on a particular tissue or organ, we call it benign tumor or localized tumor. But if it is developed or if it spread around other part of the body or most part of the body, it's no longer benign but malignant. Okay, so we've been able to note the origin of genetics, what genetics mean, and its relation called the genomics, what they mean. Yes, we want to see some important terms we need to know concerning this topic, genetics. and one of it is called gene. We have been hearing this gene before now. We have been hearing this gene before now. Yes. Gene simply means a piece of deoxyribonucleic acid that provides a set of instructions to a cell to make a certain protein. Now, DNA is known as deoxyribonucleic acid, which contains the genetic instruction in all living organisms. It's important to know that it has four bases. The four bases are you talk about guanine, thymine, we'll talk about cytosine, so it has four bases: guanine, thymine, cytosine, and adenine. Yes, it's important to know that the way, the arrangement, the sequence, the order of arranging these four bases, or the sequence, the order it is being arranged, the order, the sequence, the arrangement of these four bases determines the information this DNA will carry. Yes. Now, in DNA methylation is another term we need to know, because this simply means a chemical addition to a piece of DNA to turn it on or off. Yes, we've been able to establish fast now that whenever there is sudden change in the gene, in even one of the gene we have in the body, it can result to outgrowth of tumor. And if that tumor is developed on one part of a body or tissue, maybe on the breast, for instance, if the tumor is developed on one part of the body, or one part of the organ, or one part of the tissue, we'll call it a localized tumor or benign tumor. Then we also said that if that tumor spread around other parts of the body, it's no longer tumor, it's now a cancerous cell, we call it a malignant tumor, it's no longer localized. It's important to know that if that change we talk about, sudden change, takes place within the egg and sperm part, that is, if it takes place within the egg and sperm, we'll call it a germline mutation. But if this sudden change takes place in the body cells, we'll call it somatic mutation. So we've been able to establish facts that mutation can occur in any gene, in any part of gene, and once it occurs, it will result to outgrowth of tumor, and it can be localized. The tumor can be on one part of the body, it can be treatable. Most times surgery is one of the best way of treating it. You cut off that part because it is at a particular point of the body. You cut it off so that it will not spread to other part of the body. But once that tumor has spread around the whole part of the body, it's no longer a tumor. Now it will be called a cancerous cell, we call it a malignant cancer or a malignant tumor. It's now a cancer. Okay, so it's malignant. In such case, you don't solve it by surgery because it has spread around the whole body. We also said that if the mutation or sudden change occur in the eggs and sperm, we'll call it germline mutation. But if it occur on the body cells, we'll call it somatic mutations. Now, another term we want to look into is chromosomes. Chromosome, it contains a single long piece of DNA with different genes. Yes, it is important to note that human beings have 23 pairs of chromosomes. Each pair of the chromosome has two chromosomes. That is, it got a particular person, an individual that has 46 chromosomes, he got 23 from the father and 23 from the mother, making it 46 what chromosome. Yes, that is another point. Remember, we say the 22 pairs are called the autosomes, while the remaining one pair, which is sex chromosome XX or what YY, the remaining one pair will be called sex chromosome XX or what YY. Then the next thing we want to explain is traits. We said that traits is a distinguishing characteristics that are inherited, such as eye color or leaf shape. Yes, it's a distinguishing characteristics that are inherited. What we inherit from our parents, we call it what? Traits. Okay? Now, heredity. This is the passing of genetic material from parents to offspring. Please don't make mistake or don't think that heredity means genetics. Remember, this is the passing of genetic material from parents to offspring. But in genetics, you are studying how the genetic material are being transferred from parents to offspring. So genetics is a study, while heredity is the passing of that genetic material from parents to offspring. We need to understand the gist. Then next is allele. But before we go to that allele, it's so important to note that it's possible for you to see a child. It will not be replica of the parent. It's possible for you to see a child. You look at papa, it doesn't resemble papa. You look at mama, it doesn't resemble mama. You check DNA test, it shows that the father is responsible for that, because every blood of a child comes from the sperm of a father. Every blood of a child comes from the sperm of a father. That is why when you go to DNA test, what is checking is if the father is responsible, because it is his sperm that will be responsible for the word baby. Okay, now you notice that it's possible you see a child, then you look at the father, it doesn't resemble father, maybe facially or physically. You look at the mother, it doesn't resemble mother, and you will be thinking that maybe something happened behind. No, it's possible that if you check the grandparent, the child might be a replica of the grandparent. That is why genetics is a broader topic. Okay, so it's possible for a gene to be transferred from the grandparent to the what? To a child, or to a grandchild. So it's also possible for you to see a child, he does not have anything at all, even from the father, from the mother, or from the grandparent. You could not notice anything they have in common. That takes us to individuality of what? An offspring. It can happen, because in the field of genetics, many things can happen, and it is when you study it closely, you see that child, it doesn't have anything in common with the father or the mother or the grandparents, so to speak. It is when you look or check out the characteristics of that child, you will see one way that maybe attitude, it might resemble them, but maybe facially it did not, but attitude it might. So a lot of things can happen in genetics or in heredity as well. Yes, a lot of things can happen there. When a child do not resemble daddy, do not resemble mommy, check the grandparents or the extended family. Know if the grandparents, if you resemble or maybe resemble one of them, maybe the daddy or the mommy, is possible. But if also it did not resemble any of them, and you check the DNA test, it's showing that the father is responsible, that takes us to individuality of pressure of a offspring, that such case could happen because this is genetics for you. Okay, so we'll go to allele. In allele, we say that it is a version of the gene that is present. Each person has two allele for each gene, one from each parent. If the allele of a gene are same, we say it is homozygous to that gene. Then if the allele are different, we also say it is heterozygous for the gene. So allele of a particular gene can be different, that is heterozygous. It can be same, that is what, homozygous. We also need to note about this, that each person has two allele for a particular gene. When the allele of a particular gene are same, homozygous. When the allele of a particular gene are different, that is heterozygous. Yes, we've been able to know that in human body it contains 23 pairs of chromosomes, and these 23 pairs of chromosomes goes this way, that 22 pairs of the number chromosomes are called autosomes, and we say it's autosomes. Then the remaining one pair of chromosome, which is sex chromosome, is XX or what, YY. Is XX, or please not YY, is XY. Please, I just want to correct it. It's a mistake. It's a slip of tongue. Please, the 23 pairs of chromosomes we have, the 22 pairs are called autosomes, and the remaining one pair is noted to be what? Sex chromosome XX or XY. Please have it in your mind. Yes. Now we go to another term we want to define, which is homozygous. We said that it simply means two of the same allele. When we have two of the same allele, which could be AA, they will have to be in the same case. AA, capital A, small letter A, small letter A, capital letter B, and small letter B. It could be like this. So it means it's talking about homozygous. But when we talk about heterozygous, it means we have two different alleles. That's capital A, small a, capital B, small b. You can see this well when we talk about allele for purple flowers and allele for white flowers. You see, we are talking about color of flower, but they are different color, purple and white. So you see that there are two different alleles, purple flower and white flower. So when you are talking about allele for purple flower, allele for white flower, comparing the two, we are talking about heterozygous because they have two different words, alleles. Then next thing we want to define is genotype. It is the genetic makeup of specific gene. It is genetic makeup of specific genes. When we talk about genotype, it means it is referring to the allele that a person has for a particular gene. It is referring to allele that a person has for a particular gene. That is genotype. Now, phenotype, it is the physical characteristics or appearance or traits of an individual. Okay, the physical characteristics or traits of an individual. We will talk about dominant. Dominant is simply allele that is expressed when present. That is, when a particular allele masks another allele. You say that allele that masks the other allele, that override the other allele, we'll call that one dominant. Then that allele is mask or it override, we call that one recessive. Then note this point, that dominant disease can be caused by only one copy of a gene with a DNA mutation. Then from this statement, what we mean by this statement is that If a parent has a disease, each child has 50% of inheriting that disease. If a parent has a disease, each child has 50% of inheriting it. And that is why, while assessing a patient, the patient is asked of his family history. Yes, it's one of the information we get from the parent. You ask, do your daddy have this disease when he was alive, or does he have this disease? Do your mommy have this disease? What of your grandparents? Does it, let's say now we have a diabetic patient. What we'll be checking is, okay, do your daddy have diabetes? Is he suffering from diabetes? Do your mommy have diabetes? Disease like hypertension, we do that. We question the patient on this to know if maybe he inherited it from the parent. Yes, it's also possible that someone parent do not have a particular disease, but the person develop it all of a sudden. It's possible that environmental factors can come into play in that regard. Yes, someone who take excessive of alcohol, who smokes, who take tobacco, you notice that such person is risking, is trying to expose his or her health to certain diseases. Take for instance, someone who is taking excess alcohol. It's true that in the field of drug, alcohol is a drug. Is one of the minor tranquilizer we have. It has anxiolytic effect. It can make one to sleep. Yes, we know that it depresses your CNS. We know that. But when one take it excess, what is the person trying to? What is it trying to do to the liver? You see, it's trying to bring more stress, more work for the liver. Such person is exposing himself or herself to having diabetes one day if care is not taken. So you now see that environmental factor, social life, the lifestyle of the person can come into play, and it can make it possible that a disease that the parents or the grandparents do not have when they were alive, or when as they are still alive, it will make it possible that the person will not have it because of the lifestyle. So we now see that if a patient or if a person has a disease, each child will have 50% of inheriting disease. Then we also have what we call epigenetics. That simply means the study of changes in phenotype caused by something other than underlying DNA sequence. Epigenetics. So we have noticed that it's possible that a disease that a parent or grandparent or all the ancestral do not have when they were alive, it's possible for a patient to develop it. Why? Because of environmental factor, excessive alcohol, smoking, lifestyles the person indulges in herself. So we have to be very cautious on that. It is important to note that we have three basic areas of genetics. One, Mendelian or classical or transmission genetics. Two, molecular genetics. And three, population genetics. It is also important to note that in molecular genetics, it shows in the individual's physical appearance. It's also important to note that in molecular genetics, that all features an individual is carrying, is as a result of the amino acid that individual possesses. Yes, if an individual possesses 2 glutamic acids, it means that is likely to have a genotype. Then if an individual possesses a glutamic acid and a valine, amino acid. It means that it's very possible it will possess AS genotype. Then if an individual possess valine and another valine, that is two valine amino acid, is very possible that the person will possess SS genotype, that is sickle cell patient. It will be a sickle cell patient or sickle cell individual. Okay, so you now see that all features an individual is carrying is dependent on the amino acids he possess. Okay, now in population genetics, it deals with how the genes separate in our environment. It deals with how the genes separate in our environment, such as thumbprint pattern, hemoglobin structures. They are examples of population genetics, what we study. When we say we are studying population genetics, we are studying thumbprint patterns. We are also studying hemoglobin structures. So they are examples. Okay. Now, as we introduce the topic genetics, we said that this father of genetics, Gregor Mendel, uses a garden peel, a species of garden peel called Pisum sativum. He uses a species of garden peel called what? Pisum sativum. And we said, as this video continue, we see reasons why this scientist or this father of genetics choose to pick or to select Pisum sativum. One of the reasons why he chose it is because the plant was easy to be cultured in open ground. It has short growth period and life cycle. It has constructive heritable characters or discontinuous characters. It can produce fertile hybrids on artificial pollination. The plant has perfect or self-pollinating flowers of peculiar structure that it contains both male and female parts. That is, it contains both male and female parts. So, it is diploid. That is another important reason. Then, another important reason is that the plant can be easily cross-fertilized. And the plant has true or pure breeding strains that can be obtained from local seed merchants. You can now see the reasons why this father of genetics decided to choose Pisum sativum from a species of what? Garden pea. Okay? Yes, we want to now see why was Mendel successful. Yes. One of the reasons is because he analyzed the reasons of failure of his predecessors and wrote down the program of his hybridization. He didn't just start running his experiment, he analyzed the reasons of failure of his predecessors before commencing on his own. Again, he carried out preliminary investigation in order to obtain familiarity with the experimental organisms. He studied the inheritance of one character at a time, unlike his predecessors, who had considered the inheritance of an organism as a whole. He carried out his hybridization experiment to F2 and F3 generation. In the example we see in this video, we will see how he did that. He grew different pure lines in separate garden plots, preventing chances of cross-pollination. He experimentally produced and maintained genetically pure parent plants, that is, pure breeding lines. He tested the purity by self-crossing the progeny for several generations. Number 7, he meticulously kept the statistical records of all the experiments and analyzed them carefully. He studied 7 unique characters which are visible and were represented by 2 contrasting forms or traits. Now, the 7 unique characters he talked about were seed shape, cotyledon color, silk coat color, ripe pod shape, unripe pod shape, flowers position, and stem length. Now, the two contrasting traits he crossed, round and wrinkled. When he talk about C shape, we talk about round and wrinkled. He crossed round with what? Wrinkled. Then he also crossed yellow and what? Green. That's cotyledon color. He also crossed yellow and green. That is for silk coat color, or green and white. Inflated and constricted. When you talk about ripe pod shape, then for unripe pod shape, he crossed green and yellow. Then for flowers position, he crossed axil and what? Terminal. And for stem length, he crossed tall and short. We want to see example of how the crossing was done by Mendel. We want to see one of the examples of how he did the crossing and the interpretation or conclusion he came up with from the crossing. Now let's see example of how this scientist Mendel did his crossing. Now we'll pick plant height as the character we want to what? Cross. After picking it, after our crossing, we will see conclusion that we could get from that, and that was the conclusion he too got. Now, under plant height, from the table, from the trace we showed in the table, we said that he picked tall and short. Tall and what? Short. He represented tall with capital T and short with what? Small letter t. Then you will notice that when you cross this, it will be giving you capital T, small letter t, capital T, small letter t, capital T, small letter t, capital T, small letter t. That is all will be what? Tall. He realized that this F1 generation are all tall. So this is the gamete. This is the gamete. Gamete, F1 generation, they are all what? Tall. Now that means as this gamete and this gamete met together, they produce all tall offsprings. Now he now pick one of these two of these offspring we have here. That is T, T, and T, T, and cross them. We have T, T, we have T, T, then we have T, 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 T. So when you now cross this also, you now realize that this trait, this gene TT, this trait we have here now, which shows here, it didn't show here. It was dominated. I mean, it was, the effect didn't come out here. He now called this one to be recessive gene, because the effect was shadowed. It only reflected in the F2 generation. It only reflected in the F2 generation, but in F1 generation, this tallness shadowed it. And this is a trait for what? Short. Meaning that in the F2 generation, the parents were able to get a short offspring in the F2 generation. But in the F1 generation, they were all tall. That means this short offspring, or this offspring that is short, was override were masked. Their effect were masked in the F1 generation. This tallness dominated them. Then this is what? These are all what? Tall. And these are dominant. They are dominant what? Gene. That means he said that the ratio of dominant to recessive, that what he realized is that ratio of dominant to recessive is 3:1. That is dominant to what? Recessive. He also came up to say that the phenotypic ratio of this crossing is 3 tall is to 1 what? Short. That is the phenotypic ratio, the physical appearance. The physical appearance he got here is 3 tall and 1 short. Then the genotypic ratio, he realized that he has 1 big tall, 1 TT, then 2 TT, 2 capital T and small t, and what? 1 short. That means this is the genotypic ratio he got from his what? Analysis. Now it is on the basis of this result we just got that he made the following conclusions. 1. He said that since the original parental stocks were true or pure breeding, the tall variety must have possessed 2 tall factors TT, and the short variety must have possessed 2 short factors small t. Then the F1 generation possessed 1 factor from each parent, which were carried by their gametes. Then these factors do not blend or mix in the F1 generation, but retain their individuality. Also, in the F2 generation, both of alternative factors that define the parental lines appeared. The character that appeared in the F1 is dominant over its alternative, which is recessive. You know that in our F1 we got TT all through. It said it is dominant over the alternative, which is recessive, because if this small letter t is dominant, it's supposed to have reflected in the F1 generation, but it did not. It reflected in the F2 generation. F2 generation will look like it reflected in the grandchild or the grandchildren they have. So that is what recessive. That character is recessive, because if it is dominant, it's supposed to reflect in the F1 generation. Also, he concluded by also saying that in the F2, the factor or character that appeared in the F1 to the character of its alternative was found about 3 as frequently as its alternative. You recall when we talk about phenotypic ratio, we say it's 3:1. Okay? Then, from all this, he delivered his first law. He made known his first law, which we call principles of what? Segregation. And the law states that the characteristics of an organism are determined by internal hereditary factors, which occurs in pairs. Only one of a pair of such factors can be represented in a single gamete. I repeat, the characteristics of an organism are determined by internal hereditary factors, which occurs in pairs, and only one of a pair of such factors can be represented in a single gamete. To put it in a simpler way, the first law, or Mendel's first law, simply says that the members of a pair of alleles segregates or separates clearly from each other during the formation of gametes. We'll be stopping here for today's class. We will see more videos on genetics on our channel. Thanks for watching today's video.